Today, we're very excited to have Xin Chen with us today. She's a PhD at UCSF. And the title of her talk is Differential Roles of Human CD4 plus and CD8 plus regulatory T cells in controlling self-reactive immune responses. And just a little bit about her. She completed her undergraduate studies in biology at Case Western Reserve University and earned her PhD in Dr. Th1nk's lab at the Jackson Laboratory, where she investigated the role of glucose metabolism in IL-17 secreting cells. She's currently a postdoctoral scholar in Dr. Mark Davis's lab at Stanford University, focusing on immune system regulation in autoimmune and infectious diseases using human organoid systems. And I will drop her paper into the chat for those who are interested in taking a look. Thank you so much. Welcome. And we're excited to hear more about your work.
Thank you. Thanks for the introductions and invitations. I'm here to share our new papers on the studying role of different regulatory T cells in controlling self-reactive immune response using tonsil organoid. So animal models has been a very useful tool to study generating the knowledge of basic immunology. So in fact, almost all our knowledge from the immunology textbooks is using animal models. However, we realized that mouse and humans eventually are still different. So the biggest difference is that human has a much diverse genetic background compared to mice, which they normally produce as the universal genetics background. And the second difference is that human has a more complicated history of infections. And also, besides, there's a difference in the microbiome. So all of those are very important to, in terms of inducing immune response to different antigens. So one example from the vaccine studies is that all this promising vaccine that works very well in mice, it doesn't work in humans. So the group at Stanford University, led by Dr. Davis, spent like decades and a lot of money to try to work out a good model to enable human studies. So we realized that we need a studied human immune system directly to discover just what is affected. So we come out a good system using the human tonsil organoid that we collect the tonsils from patients with sleep apnea. And this is a very normal surgery. So every day is discarded, the tonsil is discarded every day. But if you collect it from the surgery room, and they bring back to the lab, and they mechanically dissociate it, which means you can cut it up into small pieces, and then go into like single cell suspension. And then we culture them into transwell at a very high density. And then we stimulate them with some antigens, for example, live attenuated flu vaccines. And after 2 and 3 days, the cells start to get cluster. So for example, this is the vaccine stimulated organoids compared with the unstimulated culture. So I think the most valuable things in the tonsil organoid system is that they contain a critical structure for germinal center reactions, so the like zone and dot zone, which is important to produce antigen-specific antibody. And it's also important for the process of somatic hypermutations and affinity maturation, B-cell differentiation in response to flu vaccine. So when I joined the lab, Mark threw me an important question. So how we can maintain a tolerance to self-antigens while inducing a vigorous response to foreign antigen, which is always a central question in immunology. So currently, we know that there's a very well-famous regulatory T cell, which is CD4 T cells, that they express transcriptional factor phosphate 3. They through a different pathway that can control the autoimmunities, like through cytokine inhibitions, inhibit the activation of T effector cells, and some metabolic disruptions, or like consuming IL-2 or through some cytolytics. Well, we recently, there's another type of regulatory T cells we call CD8 Treg. They are recently identifying they might also play an important role in autoimmunity. So this CD8 T cells express KIR, is a killer cell receptor in human or LY49 in mouse, that they play an important role to control the autoimmunity while we have infections. So if there's two different types of regulatory T cells that might affect it in human, what are their relatively role? So why we need two regulatory T cells? So that's what my first postdoc project. So in order to do that, we know that so for the function of CD4 Treg, if it is the expression of phosphate 3, so this is important for the stability of the CD4 Treg. And this CD8 Treg, there's important genes encoding the granzymes because they're cytotoxic cells. So we try to knock out these two critical genes for their functions, and then study what happened in the organelle after the knockout. So in order to generate the phosphate 3 or granzyme B knockout, I enriched the T cells from the tonsil single cell suspensions. And then I use electroporations and together with the Cas9 nuclear ribosome protein in complex with the targeting guide RNA. And then I combine the rest of the cells with the modified T cells, and then I culture them into the transwall. So first thing I check is what are their knockout efficiency, because it's still challenging to get a very good knockout efficiency from the primary T cells. So throughout all the modification protocol, we demonstrate that we can knock up to 95% efficiencies for the phosphate knockout in CD4 Treg, and also almost a 95% knockout of KIR in the CD8 Treg. In addition, we find that in this knockout organelles, they also display some pro-inflammatory phenotypes. For example, I can see some activations of CD4 T cells and also increased percentage of T reticular helper cells and CD8 T cells in both knockout. So in terms of B cells, I do see some B cells activations and B cell differentiations in both phosphate knockouts and granzyme B knockouts. As you can see, there's increased percentage of germinal center B cells and plasmablasts, which is a terminal B cell that can produce antibodies. So while I can see some activation and proliferation phenotype in both knockouts, however, I couldn't detect any autoantibody productions. And then we go back to some paper and also in some mouse study, and we find that because it's known that the infections can be the primary factors that can induce autoimmunity. And also in some mouse study, they also find the production of autoantibodies produced after the mouse have some LCMV infections. So we try to repeat this experiment, but in fact, the organelle with the live attenuated influenza virus. And together, also we stimulated with some autoantigen pool, including the proteinase 3, PR3, core histone, double-strand DNA, and some small nuclear ribosome nuclear protein. So those are the common proteins that common autoantigen that we can see from the autoimmune disease patients. And then I use the ELISA assays to measure the autoantibody production. So in the wild type organelle, even though with the activations, with the simulation with live attenuated flu vaccine, or together with the autoantigen, I didn't see any increase in the production of autoantibodies. However, in the phospho-3 knockout, which I disabled the function of CD4 Treg, I can see a slight increase of autoantibodies after stimulation with live attenuated vaccine. And this productions is even become higher if you stimulate it with LAIV and autoantigens. And this is very similar patterns in other other autoantigen specific antibodies. However, if you compare with the Granzyme B knockout, which we disable the function of CD8 Treg, I didn't see a significant increase with the stimulations. So this indicates that the CD4 Treg are the major regulator of autoantibody production. And if we go back to the patients and then we check for their gender, and we notice that for those organelles that with higher autoantigen specific antibodies are mostly derived from females, which indicates that the female donor tend to produce higher level of autoantibodies than males, which is consistent to what we normally see from autoimmune disease. So once we find that there's high level of autoantibodies, so we would expect that there are also expansions of autoreactive B cells. So in order to probe that specific autoreactive B cells, I modified reagents that just developed in our lab. It's a multimer. It's similar to tetramer, but it has a much. It's like a 12 peptides peptide specific probe that can bind a very sensitively bind some antigen specific cells. So we call it spheromer. So using spheromer, I find that there's a significant increase of autoreactive B cells in the phospho-3 knockout organelle. However, I didn't see a significant increase of autoreactive B cells in the Granzyme B knockout, which consistent with what we normally see from the autoreactive antibodies, that there's much higher level of autoreactive antibodies in phospho-3 knockout organelle. So what so if you remember back that we see even though we didn't seek an increase of autoreactive B cells or autoreactive antibodies in the granzyme B knockout, but we do see some expansion of the T cells in the granzyme B knockout. So so another hypothesis is that maybe this CD8 Treg will the major effect of CD8 Treg is on the T cell side. So that's what we also generate a experiment and then measure the percentage of autoreactive CD4 T cells in all these organoids. And interestingly, we find that in the granzyme B knockout, which I disabled the CD8 Treg, after the stimulation of flu vaccine or the vaccine together with the autoantigens, there's significant expansion of autoreactive CD4 T cells, which indicates that the CD8 Treg might be the major controller in regulating the autoreactive T cell. So, and also consistent with what we normally see from the antibodies. So those B cells that have the highest expansion, those cells have high expansion is also derived from the female. Okay, so because we know that the CD4 Treg also express granzyme B. So in order to eliminate the possibility that we also disable the function of CD4 Treg in this granzyme B knockout organelle, I knock out. So I did a knockout for all the conditions, while I also deplete the CD8 Treg by using the antibody depletion methods. And then I'd add back the killer positive CD8 T cells, or I add back the killer CD8 negative T cells. And then we find that so with the granzyme B knockout, as previous data shows that there is an expansion of autoreactive CD4 T cells. But if you add back the CD8 killer, it looks like this CD8 killer CD8 T cells suppress the expansion of autoreactive CD4 T cells, while the CD8 negative, the killer's negative CD8 T cells does not. So this is the evidence that this granzyme B, so this killer CD8 T cells can control the expansion of autoreactive CD4 T cells. We also have a control the using experiment that determine specific to foreign antigen, which is the H8 molecules from the flu vaccine, and we didn't see a significant response, which indicates that this CD8 killer T cells specifically target the pathogenic autoreactive T cells. And lastly, one interesting observation we find that from the phospho-3 knockout organoids, they can produce antigen-specific antibody with higher binding affinity. Well, there's no significance in the granzyme B knockout organoids. And this data remind us another paper with the air deficient patients, which they also have some autoimmune phenotype, that they find there are also some high affinity antibody related to foreign antibody from humans. So that indicates that with our genetic modifications in the tonsil organelles, it can also recapitulate what we can see from the patient with autoimmune disease. So lastly, since we have these reagents, we are able to detect, we are able to measure what would be the percentage of autoreactive cells in the body. Because originally from the mouse model, we think that all the reactive cells should be deleted before they enter the peripheral tolerance. However, it is not the case in humans. And also, so we do find some percentage of autoreactive B cells in the healthy donor, and this is much higher in the female donor. And also we see a very strong correlations in related to age. So the elderly, so all the organelle that derive from the older donor will have a higher level of autoantibodies induced by the flu vaccines. So for the conclusions, we demonstrate a genetically modified tonsil organelle to recapitulate some autoimmune phenotypes, including sex bias, and we investigate the relative contribution of Tregs to control self-reactive T cell and antibodies in humans. And we particularly we find that the CD4 Treg that express phospho3 are the key trap point for high affinity antibodies as well as the production of autoantibodies. While the CD8 Treg, which express KIR, have a higher control of autoreactive T cell response than CD4 Treg. Therefore, we think that the CD4 and CD8 Treg have a distinct but also complementary roles in regulating cellular and humoral response and preventing autoimmunity. So I want to thank Marks for his mentor and all the lab members for the help. And I also want to thank some Dr. Capasso for providing the tonsil, and also thanks for donor Network West for providing all the resources, and also thanks for the fundings, such as HHMI and Gates Foundations for that. Yeah, thank you.
Thank you, Xin. This was really interesting. I'm just opening it up to anyone who's in the audience who'd like to ask a question. I had a couple of questions. I guess I would say one of my first questions is what follow-up studies are planned to further elucidate the mechanisms of the Treg mediated immune regulation?
Yeah, there are a lot of directions. So one we interested is since we already know there are some autoreactive cells, especially autoreactive B cells in healthy donor. And we luckily we actually have some donor with SLE history. And with the spheromer, we find there are like almost a 20-fold increase of autoreactive B cells. So we want to determine what are the alternate in terms of like transcriptional level, transcriptomic level, what's the difference between the autoreactive B cell in the healthy versus in the patient with disease, and how is there any pathways that will turn this autoreactive B cell into a pathogenesis of autoimmune disease, and then we can. So once we identify any like genetic markers, we can use the knockout technologies and to study the mechanism with the organelles.
That's really fascinating. Any other questions? Anyone else?
Yeah, I have one, please.
Oh, yes. Hi, Sylvain.
Hi.
Yes, I'd love to have a question from you guys.
Yeah, hi. Thank you for this presentation. I have a question regarding your essay where you show the efficacy of KIR positive CD8 T cells regulating the CD4, the autoreactive CD4 response in the granzyme B knockout organoid. So you say that only the KIR plus CD8 T cell were able to reduce the number of autoreactive CD4 T cells. But in this assay, in the same assay, did you also test the Tregs just to compare the capacity of the Tregs, the classical CD4 FOXP3 Tregs, to modulate the autoreactive CD4 T cells in this very same assay in the granzyme B knockout environment, just to compare side by side?
We do look at I just look at the I didn't do the functional assay, but I checked the percentage of FOXP3 in the organelle, and I didn't see significant difference. Even though but after infection, the FOXP3 is slightly decreased in some of the cases. But in terms of like adding in KIR KIR positive cells versus adding in KIR negative cells, the FOXP3 percentage doesn't change a lot.
Okay, I was wondering whether if you remove the Tregs as you did for the KIR, and then you add them back as you did for the KIR plus or the KIR-NEC CD8, I mean, would they be able to inhibit the autoreactive CD4 T cells less or better or in a similar way as the KIR plus, just to compare their suppressive function in the very same assay? Yeah, this was just my questions.
Yeah, I didn't do that. We didn't do the depletion with CD4 Treg. The reason why I do knockout is so one thing interesting is that in the tonsil, so we always think that the CD4 Treg has very high CD25, right? This is a marker. But in the tonsil, they're almost half of the FOXP3 positive is CD25 negative. So that's why I cannot deplete the cells by like FACS. I can only knock out the FOXP3 to try to disable them, and then but then we did the experiment with knockout FOXP3, and we didn't see a significant increase in autoreactive T cells, so we just end it there.
Okay, thank you.
Thank you for the great questions. Okay, I just, thank you again, Sing. It was really great having you on, and we look forward to more information coming from the Davis lab regarding this new system, this organoid system derived from tonsils, and it's exciting times. A lot of great things coming out of Stanford in the last week or so. So we've got our eyes on all of them. And thanks again for sharing your really interesting work with us today. Hope you have a great rest of the day.
Thanks too.
All right, bye-bye.
Thank you. Thank you very much.
Thank you.